going from there. All right. So good afternoon, I'm Scott Stauble. I would like to welcome you to the concurrent session on microbiology part 2. In this session, we're going to have about 6 presenters. The first 5 are going to present kind of in one group, and then a 6th presenter at the end to share their research while in the START program. We will begin with a group that's been working with Professor Greg Johnson. It's Malik Abdeen, Salma Al-Kafaji, Karine Yeya, Noor Mahareb, and Lisbeth Rezendiz Alas. So we'll start with those students. We will be recording this session, so you're welcome to turn off your camera if you don't want to be recorded. Keep in mind to keep your microphones off when not speaking. We will have time at the end of presentations for questions, so if you have any questions, feel free to type them in the chat. We've got Kathy Jordan, one of the associate department heads for math and physics, monitoring the chat for any questions. And at the end, if you want to unmute and ask your question, you'd be welcome to do that as well. So we'll go ahead and get started with a video recorded by this first group of students, and then they will present on some of their findings after that. All right. Nope, that's not the right video. Please hold. Technical difficulties. All right. Is everybody able to see that video?
Yes.
Yep.
Yes.
Hey everyone, my name is Karine, and I'm gonna start off our meeting by introducing our project. Our project is about addressing the growing problem of antibiotic resistance. The antibiotic resistance is when a germ, like a bacteria, becomes immune or resistant to the antibiotic. In this project, we were very cooperative with our mentor, Mr. Johnson, to have better results. We should able after this project to understand why the antibiotic resistance will help us and how the soil samples will help us determine which produce a chemical entity for the development of their resistance. Our goal after this work is to gain an understanding of the antibiotic resistance problem facing the world health. We used the biosafety level 2, the BSL2, in our lab, and we learned the protocols of the small world initiative, to be able to apply techniques in the lab to discover new chemical entities. So why is it important to follow small initiative word technique? So the small word initiative, is a novel curriculum that inspire students to seek science careers while discussing a global health problem. This one-of-a-kind class approach integrates the potential of constructive learning to accomplish academic and scientific objectives. Students from all over the world extract the bacteria from soil in their local habitats, which could contribute to the creation of new antibiotics. This is especially important given that soil bacteria and fungi are the source of more than 2/3 of antibiotic. The Small World Initiative biology course distinguish itself from conventional courses by offering original learning opportunities rather than focusing on just notes and books and tests with preset outcomes. Students gather a soil sample, extract diverse bacteria, monitor their bacteria against clinically important microorganisms, and sourcing antibiotic research by using the intellectual resources of many individuals working on a global issue at the same time. Hey everyone, my name is Karine, and I'm gonna start off our meeting by introducing
resistance are prevention.
sourcing antibiotic research by using the intellectual resources of many individuals working on
Report. CDC
Gonna get this working, hang on.
issue at the same time.
CDC posts reports annually on antibiotic resistance, and I'm focusing on their 2019 report. And some strategies that they mentioned that we've developed over the years that work to combat antibiotic resistance are infection prevention in long-term care facilities. So this includes nursing homes and elderly homes. Preventing device and procedure-related infections. So this could be in the surgery room or OR, using more sterile technique with cleaning equipment. And also safer food handling. So some statistics that I thought were very important to mention with how far we've come is that we have 80% fewer deaths from antibiotic resistance overall since their 2013 report, and 28% fewer deaths from antibiotic resistance in hospitals since their 2013 report. These are large percentages, and we have come a long way. However, even though we've come a long way, there's always room for improvement, and there are still challenges we're facing in terms of antibiotic resistance. Some aspects we need to work on are stopping the spread of germs in animals, and we need to focus also on threats from outside the US, like bringing in new diseases from other countries. And we also need to improve antibiotic resistance all around the world. So if we work on improving antibiotic resistance all around the world, that would also minimize threats that we experience in the US. So despite how far we've come, we have some statistics that are kind of more drastic about how antibiotic resistance affects so many people. So we experience 2.8 million plus antibiotic resistant infections each year, and we have 35,000 plus deaths from antibiotic resistance each year. And these statistics show why it's so important that we work to combat antibiotic resistance and continue to do antibiotic resistant research.
I don't know why this video is not working that well for me. It just doesn't want to keep rolling.
Other countries, and we also need to improve antibiotic resistance all around the world. So if we work on improving antibiotic resistance all around the world, that would also minimize threats that we experience in the US. So despite how far we've come, we have some statistics that are kind of more drastic about how antibiotic resistance affects so many people. So we experience 2.8 million plus antibiotic resistant infections each year, and we have 35,000 plus deaths from antibiotic to combat antibiotic resistance and continue to do antibiotic resistant research.
Yeah.
It just doesn't like me.
Okay, guys, this is something
Yeah.
that you got to learn. It's another learning experience for you guys.
Yeah. So I think
When technology gets a little goofy on us, we jump in and we just go ahead and continue on our own and we talk. So I'm not sure who was going to talk next. It looks like there was in a transition there, but I'm sure which one of you might want to talk next about the procedure. So let's get right to the procedure, exactly, what we did, or at least in summary. When you guys want to take that? Corinne, you already talked in it. Noir,
did you guys try downloading the video? Maybe that might help. But if that doesn't work, we can, and if we're running short on time, we can try to say it, I guess. But
yeah.
I'm downloading it now. A good point. See if I can make that work. All right. Open. Right. Pause that. Share screen. One. We were.
We work to combat antibiotic.
Oh, well, there's our problem. The video gets corrected.
As the temperature, the time acting async.
Okay.
There, but it looks like it recovers.
Yeah, it looks like it could go in there.
Into a 4.5 millimeter solution of phosphate buffered saline. The soil was then diluted in a serial dilution at a 1:10 ratio. Elanocyclohexamide of 0.01% was also made onto 3 separate laurel broth agar plates, and it was 3 different agar plates because we designated one for the 10-2, one for the 10-3, and the other for the 10-4 dilutions. We added 3 drops of these separate dilutions onto each plate and then spread it out throughout the plate. The 3 separate agar plates were then incubated at 30 degrees Celsius for approximately 1 to 2 days to allow for maximum growth. 30 to 50 colonies that were uniquely distinctive morphologies were then isolated and plated onto a gridded laurel broth agar plate. We gridded the plate to have approximately 50 different individual squares, so then each colony was individually planted on each designated square. After this, we reduced the amount of colonies that we were going to test against, and we reduced it to around 20 to 30 different individual colonies that we then selected and placed onto a gridded laurel broth agar plate and isolated it into that separate agar plate. After this, a line of the Small World Initiative bacterial strains were then made. The bacteria strains that we tested against will include Pseudomonas aeruginosa, Arwenia carnivora, Enterobacter aeruginis, Bacillus subtilis, E. coli, and Staphylococcus epidermis. After this, we had made grids of approximately 20 to 30 spaces, which we drew onto the back of the agar plates with the lot of the different bacteria strains that we were going to test against. The 20 to 30 colonies chosen were then transferred onto each of the laurel broth agar plates with the lot of the bacteria strains using toothpicks, and All of the agar plates were then incubated at 30 degrees Celsius for approximately 1 to 2 days, again to allow for maximum growth. The unknowns were then streaked for isolation if the colony demonstrated activity against the tester strain or if there were any indications of inhibition of growth, which allowed us to know if it was having any activity showing antibacterial resistance. If the activity was further confirmed, then we begin the identification of the unknown stage.
Seems like we may not have sound for this portion.
I can try to do it.
Okay.
Procedure. Okay. Let me just pull it up. So for this third procedure, this is for the identification of the unknown. So for the identification of unknown, we used a gram stain test. So first what we did was we streaked glass with the samples that had confirmed inhibition. And before we continued on with the next step, we wanted to fix that the sample on the glass. So we first put water on it and then put it over an open flame. So when we put the stains, it wouldn't rinse out, or water, the colony wouldn't rinse out. So the second step was that we first poured the crystal violet stain and we let it sit for about a minute and then rinsed it off. Afterwards, we applied a gram iodine stain and left it for a minute and rinsed it off. We then applied decolorizer and left it on the glass for about 8 to 11 seconds. And then we then applied saffron to the glass and left it for about a minute. And then we took our stain species to microscope to identify the bacterial morphology within the dirt sample. And typically after dirt sampling, we would do sequencing of the rRNA of the strains of interest to determine the species of the organism. But unfortunately, in this internship, we didn't have the time to do that. But yeah. And the next slide is the results, which is what I'm going to do. So this is for everyone. We all did the same thing, the serial dilutions. Let me just put this on present mode. Oh, sorry. So I did serial dilutions from 10 to -2, 10 to -3, and 10 to -4. Most of us did, but some of us did, I believe, 10 to -3 to the -5th. But for 10 to -2 to the -4th, we calculated that there are about 500,000 to 1 million cultivable CFUs per mil. And according to the Small World Initiative, there are about 10 to the 11 organisms in 1 gram of high organic soil. And then I also conducted the hypothesis. So our hypothesis was, it's pretty straightforward. Let me just, that did that. So our overall hypothesis was if we take soil samples as a source of bacteria against certain infectious species, then some of the soil samples will discover organisms with antibacterial activity against the selected species. So each one of us took soil samples from any given location that we wanted, and we wanted to see if there were any microorganisms within that soil sample that would produce antibacterial properties to fight off against the tester strains. And then I think there's, after that, Nora's results, which that wasn't recorded, so our results weren't recorded.
Yeah, so
Okay, so
Nora, if you'd like to.
we're going to start.
Yep.
Yeah, I can start. Is there do I have to screen share my presentation?
I can.
Or the results?
I can screen share it. Hold on 1 second.
Okay.
There you go.
Okay. So in my presentation, I got to the part, in my research actually, sorry, I got to the part where I isolated 50 bacteria from the lawns I grew by plating them on two 5x5 grids. If you go to the next slide, you could see, yeah. So these are my two 5x5 grids that I plated bacteria on. And so basically, they're very diverse results. Like like the bacteria I plated were very diverse, and I had a lot of different morphologies. Could you please go back up, please? Thank you. So I was able to isolate many different organisms, but I was not able to test any of the organisms against the tester strains yet, and I will do that. They are viable results, and they will be moved on for further testing in the future.
I have Elizabeth next.
Hello, so my name is Lisbeth and I'm going to share my results with you guys. So our main goal was to find natural occurring organisms that produce antibiotics. So we first started by creating a lawn of various bacteria strains onto each agar plate. So I first started by planting 26 individual colonies onto each of the 6 agar plates. As you see in the picture, the first one was my stock plate, and then the rest of the 5 agar plates were the ones with the bacteria strains that we were going to test against. And I tried to make sure to make the grids and number the grids as identical to one another as much as possible, just so it keeps consistency, because that was very important throughout. After the plates were then incubated at 30 degrees Celsius for like approximately 1 to 2 days. Again, like I said in the procedure parts, I don't know if it was well heard, but for maximum growth, along with the aid of my mentor, Professor Johnson, we both selected viable colonies that demonstrated inhibition of growth. And we chose that colonies number 1 and colony number 20 appeared to be consistent colonies that demonstrated antibiotic resistance. So on the slide, you can see that the bacteria strain is followed up by a measurement, and that's the length or distance between the space of the inhibition of growth. We specifically looked at colony number 20 when we tested against E. coli because it actually performed very different compared to the others. It had formed 2 rings. The outer ring measured 4 to 5 millimeters, while the inner ring measured 2 to 3 millimeters big. And another interesting event that happened was that our number 1 unknown also had grown a red stain in the middle, and this was for our stock agar plate. If you move on to the next slide, I zoom in into a picture of that stock plate, and you can see the red stain. We didn't know if it was either contaminated or if it actually morphed into this, but we did run more tests to see what happens. And if you move on to the next slide, please. This is where we isolate the unknowns once again. And as you can see, I divided it into 3 sections because one was for the number 20 unknown, and then the 2 sections were for that red spot and then that base or original colony, since we isolated those 2 separately. It somehow turned both of them red, but it shows that colony number 1 it shows that colony number 1 had shown inhibition for E. coli, Erwinia carnivora, and Staphylococcus aureus. And for colony number 20, it showed for Bacillus subtilis and Staphylococcus aureus. Additionally, for our unknown number 20, we also observed that there were formations of rings around the colony. And although inhibition of growth was not prevalent, our findings do support the conclusion that activity is present, which impacts the growth of the colony. Now we can move on to the next slide. We proceeded by doing a gram staining on our unknowns to see the morphology, and our number 1 unknown showed that it was a gram-positive bacillus for both the red and the original. And these results basically indicated that since both strains of the unknown were gram-positive bacillus, they came from the same strand. And if you move on to the next, we see that we also performed a gram staining on our unknown number 20, where it produced a gram-positive streptobacillus with some vibrio, meaning that some of the organisms were bent or curved shaped. And currently, further identifying tests still need to be performed, such as the polymerase chain reaction or PCR, to further investigate the identity of the unknown organisms, since we did not have enough time. But if we did, that would be our preceding steps to find the identity of these organisms.
Is that okay if I share my screen?
Yes, go for it.
Yeah.
Oh, thank you. So for my results from suspensions 50 were chosen for isolating the organisms from soil dilution plates. From these 50 colonies, 25 were chosen with distinct morphologies to test against tester strains. The listed tester strains were E. coli, Enterobacter aerogenes, Bacillus subtilis, Staph aureus, Pseudomonas aeruginosa, Erwinia carnivora, Micrococcus, and Micrococcus luteus. So for my results, we took from the 50 colonies, I took 25 colonies and tested them on E. coli, Enterobacter aerogenes, Bacillus subtilis, and Staph aureus. So out of the 4 strains, 2 colonies that I labeled 23 and 25 for my unknown dirt sample showed inhibition to E. coli and Enterobacter aerogenes. Since dirt colonies 25 and 23 showed active inhibition, they were retested to confirm, and we continued testing only these unknown dirt samples on the remaining 3 strands. And so here's a picture of 23 showing an inhibition of Enterobacter aerogenes. So you can probably see the inhibition zone a little bit. And then for 25, it inhibited E. coli, and you can see it has a stronger zone of inhibition. And so I tested 25 and 23 on the remaining tester strains and got active inhibition from M. luteus for both 25 and 23. From E. carnivora, I got only 25 that showed active inhibition. From Staph aureus, only 25. And for B. subtilis, only 23 showed weak inhibition. And lastly, from P. aeruginosa, I only got 23 that showed weak inhibition. And here's a picture. So I circled the ones that did have some inhibition. So for this one, Staph aureus, you could see 25 has a little bit of an inhibition zone. And then for E. carnivora, 25 has a weak inhibition zone, but I feel like the camera, you can't really see it that well. And then 23 didn't have any inhibition. And then for B. subtilis, only 23 showed a weak inhibition and 25 didn't show any inhibition. And then for M. luteus, for both 25 and 23 showed pretty good inhibition zones, especially 25. And then for P. aeruginosa, 25 and 23 did not show any inhibition. And so I took 23 and 25 for a gram stain to further identify the microorganism. So for 23, this is a picture of 23 right here. It's gram positive and it's kind of shaped like bacillus. And for 25, this is 25. It's gram positive and it was shaped like coccobacillus.
Very good.
Yeah, that's mine.
And yeah, I was gonna say, I think Karine is next. Oh, Karine, you're muted. Sorry.
Very sorry.
It's okay.
Okay, so I'm gonna show my results from the first 50 colonies that we chose from the soil dilution plates. I chose 20 colonies to test against 4 tester strains first, which is E. coli, Staph aureus, Enterobacter aerogenes, and Bacillus subtilis. And I saw 2 colonies that inhibit first the Staph aureus, which was number 15, and the second colony inhibited the Enterobacter aerogenes, and it was number 20. But for E. coli and Bacillus subtilis, I did not see any inhibition. And then I retested the activity of the 2 colonies. So for number 15, it reconfirmed its activity for Staph aureus, but for number 20, it did not confirm its activity for erogenase, but it did show a weak inhibition for for Staph aureus, if you can see in this picture. And then we did retest for 3 more lawns, which is Micrococcus luteus, Pseudomonas aeruginosa, and Arena curativora. And number 15 did show an inhibition, and it looks like very clear. But number 20 did not show any inhibition. For Arena curativora, number 15 also showed like a very weak inhibition. I don't know if you can see it in the picture, but if you can see it on the plate, it will show a weak inhibition. But number 20 did not. It did not show any inhibition. And for Pseudomonas aeruginosa, it did not show any inhibition, neither 15 nor 20. And for the gram stains, we did the gram stains, and they are both gram positive. They look like streptobacillus because they are end-to-end in a chain, and they have rod shape. They looks the same, but they are a little different because number 15 are thinner and longer, but number 20, if you can see, they are fatter and they are more shorter. Thank you.
I believe Malak is our last person to give results.
Hello, let me.
You need me to share it for
you? Oh, can you see my screen?
Yeah, I think so. Yep.
Okay. So for my results, I took my soil sample from my front yard garden, and when I plated them onto 4 of the bacterial background lawns, I found that 2 of my organisms had inhibition, and those were 2 and 6. So I streaked those onto 6 different bacterial background lawns, and found that both of them inhibited S. aureus and luteus, B. subtilis, and E. erogenus. However, neither of them inhibited E. curativora and P. aeruginosa. As you can see in the image, the circular
No. Lost her.
Yeah, I think we lost her.
Hard while you're driving.
Yeah. She's riding, actually. I said, you're not driving.
Yes,
No,
yes.
she's riding, but would even be harder.
All right. Well, I think we are going over time as it is, so
Yeah.
I think we'll have to end it there.
Oh,
you're back. We lost you.
Okay. Yeah, we lost you.
So as you can see in these images, after putting them under microscopes, I found that both of them are gram positive, and although you could see some pink, I do believe that's because of variety of thinness and thickness on the plate, on the slide. So I do believe they're both different, and both are gram positive. They're different in morphology, and yes. So for the conclusion, there are many microorganisms in soils that make antibiotic compounds. It appears that different microorganisms have the ability to produce these antibiotics, and they're very diverse and different. So the next steps in this is to try to identify these microorganisms, and possible ways are to find gene sequencing and try to find perhaps a chemical that they produce that can cause inhibition. And I do believe it is worth pursuing more research into this. Thank you.
Very good.
Thank all of you very much for that. It was wonderful. You all have amazing results. I'm interested to see what identification can be done going forward. We do have one final presentation, And that is going to be Oliver Roper, who has been working with Mindy Miller-Cottrell. So he unfortunately was not able to join us today, so he has shared a video about his research, which I will pull up right here, and we'll get some background on what Oliver's been working on.
Hi, I'm Oliver. I'll be talking about my science communication project. Two of the most present global issues in our society today are climate change and food insecurity. Both these issues are in many ways interconnected and affect not only the environment, but also an extension to that people. A tool we can use that connects these issues and can solve these issues is the soil and its microbiome. Since the microbiome is such a powerful tool, I think it's important to communicate its many roles in the world and what the threats are to those that are making it, so we can't use them to combat these problems and eventually just end up perpetrating them instead. In my project, the goal was to effectively communicate a topic to an audience so that even if you don't have a background in sciences, you can also see how these relationships and processes in our soil affect our well-being every day, and how we need to make efforts to restore and regenerate the soil and its microbiome. In my presentation, I start by going over the basics of what a soil microbiome is and going into what it actually does, which includes interactions in biogeochemical cycles and with plants and plant health. With this basis, I was then able to talk about these threats. Here I focus more so on corporate agricultural practices like large-scale deforestation with tropical rainforest land conversions, for example, since these are some of the practices that have some of the biggest negative effects on our planet today. So from that, some solutions to these agricultural issues are adopting regenerative measures to improve soil microbiome health, to heal the soil, provide better plant yields, and to better our environment and our health. In each of these topics, I wanted to get across that these issues are rarely isolated and that we are as much a part of these interactions and relationships as the bacteria below ground are. And if we can move forward with this in mind, then we can utilize these relationships to solve global issues like climate change and food insecurity.
Excellent. All right. Well, we have gotten a good introduction into the microbiology world. We have gone a little over time, so I'm going to post. We don't unfortunately have any time for questions, but I will post the link to go back to the main meeting so we can join back in there. But thank you all so much for your presentations and your hard work this semester. It's great work. So it's good to see it. So thank you very much.
Thank you.
Thanks, everybody.
Thank you.
Bye.
Thank you.
Thank you.